Hello, everyone, and welcome to today's cryo-EM Current Practices webinar. You're joining the three NIH-funded National Cryo-Electron Microscopy Centers. My name is Christina Zamani. I'm a scientist and training liaison at the National Center for Cryo-EM Access and Training, NCCAT. I'm joined today by staff members from each center. Also here from NCCAT is Ed Eng. Joining from the Pacific Northwest Cryo-EM Center, PNCC, is Vamsi Rayaprolu, and from the Stanford SLAC Cryo-EM Center, S2C2, is Nathan Burrows. And each of them will give a quick update on the resources each center continues to make available at no cost to the research community. We'll then give the floor to today's speaker, Rachael Kretsch, who's joining us from Stanford University. Nathan will introduce her properly after our center updates. This webinar is a regular event we host the last Thursday of each month, highlighting users of the National Cryo-EM Centers who generously shared the tips and tricks that have enabled them to make use of cryo-EM in their research. You can save the date already for our talk on March 27th. Adam Oken will join us from OHSU to talk about small molecule modulation of the P2X7 receptor revealed by cryo-EM. So you can register for that already. There's a link on the events page at cryoemcenters.org. On that site, you can also find recordings of past talks and notices for all of our future talks. For final logistics today, if you have questions for any of our center staff here on the panel, feel free to reach out to them using the chat. But for questions for Rachael during her talk, please send those using the Q&A box. You can send them at any point during the talk, but we'll hold them for a moderated Q&A at the end of the talk. So let's go ahead and get started. First, I will turn over to Ed to let us know what's new at NCCAT.
Hi, I'm Ed, coming from NCCAT here in Manhattan. We offer data collection access, screening access, and training access, something that is happening in the next couple of months. We are now instituting a more streamlined logistics workflow. So every accepted proposal, their laboratory will receive uniquely coded grid boxes. And that way, if we ever receive multiple grid boxes, we'll be able to identify. So moving forward, look out for an email. We're going to give you serialized coded grid boxes. Next thing I want to mention is that we're going to be hosting a pre-meeting congress at M&M at Salt Lake City. So Claudia Lopez at PNCC, myself, and Julia Brash from cryo-ET 101 will be creating that pre-meeting congress. We want that to be a user meeting. So if you're going to be in Salt Lake City this summer, consider about applying to their PNC. If you want to find out more, go to nccat.nyspc.org.
Thank you, Ed. And I will turn it over to Vamsi.
Hi all, my name is Vamsi and I'm a microscopist at PNCC at Portland, Oregon. PNCC accepts two types of proposals, screening access and general access. Screening access proposal allows you to come on site for up to 5 days of sample preparation and screening with staff assistants. This proposal is aimed mostly at low resource institutions. General access proposal awards up to 480 hours of microscope time for 2 years. For 80 hours of microscope time a year for 2 years. That's so a total of 960 hours. This time can be used for screening or data collection. Approved general access proposals are also eligible for our training workshops, which cover topics from sample preparation all the way to data processing. Approved proposals are also assigned a scientific point of contact, a SPOC, if you may, who assist with questions about sample optimization, data collection strategies, and data processing, and several other cryo-EM related questions. Our center has an Arctica newly installed Glacios 2 and 4 Cryoses. 3 out of our 4 Cryoses have K3 cameras with biocontinuum HD energy filters, and one of our Cryos houses a Falcon 4i camera with a Citrus X energy filter. For sample preparation, we have a Vitrobot, a Leica GP2, a Vitrojet, manual plungers. In fact, we have 3 Vitrobots. If you're interested in applying, our deadline is the first of every month. We look forward to hearing from you.
Thank you, Vamsi. And Nathan, let us know what's new at S2C2. Oh, and I think you're muted.
There we go. Um, Welcome to our seminar here. We're always busy here at S2C2. We just brought online our Glacios microscope, which we're all very excited about here and are in the process of installing our Tundra. And we're continuing to plan workshops, which are available on our website. And we have our main access programs, just like the other centers, between a high-end data collection service and our exploratory screening and optimization program, as well as our training program, which we're constantly having users come visit us on site for training, which we're all excited to have you, and you can learn more about it on our website. Our speaker today, Rachael Kretsch, is one of our more active graduate students here at S2C2, and she is going to be telling you today about her projects involving RNA, which tend to be smaller molecules and definitely the more challenging samples that we experience in cryo-EM. I hope you enjoy her presentation and learning all about the different ways that RNA has been giving her challenges throughout her graduate school.
So you guys can see the right view, I am hoping.
Yeah, that looks great.
Perfect, cool. All right, so thanks for the intro, Nathan. So today I'll be talking about some, I guess, more practical examples of some structures we've solved, and how our hope is that cryo-EM will make a big impact with RNA structural biology, and that will lead to a big impact in kind of more functional RNA biology studies. So today, I'll do a brief introduction to RNA because I know it's not necessarily what everyone in this call studies, although I see some recognizable names as well. Then I'll introduce the methods that we use for RNA cryo-EM currently. And then I have two short examples, one study on some viral RNAs, and two, a study on some large bacterial RNAs. So first, an introduction to RNA. So why do we find RNA so exciting and worthy of study? I think the big reason there is that it's involved in so many processes in life. And every year we discover more and more. Some non-coding RNA does a new function, for example. So this is just a representative example of molecules that David Goodsall decided to highlight in his PDB Molecule of the Month. You can see functions everywhere from translation to very important engineering molecules, to kind of how viruses work and how bacteria are able to recognize metabolites and how many metabolites they have in their cells. But RNA structure is very underexplored relative to the protein field. So a key tool being used in structural biology and proteins right now is structure prediction tools because they've reached a high level of accuracy. So this is a plot where on the y-axis, higher accuracy prediction is up here, low accuracy prediction down here. And then we've plotted a bunch of different proteins. This is work from Cas15 competition. And then the x-axis is difficulty. So what you can see is at the beginning of the competition, which was 30 years ago now, I think, if the target was difficult, the accuracy was low, right? We had a very kind of gradual trend here. But in today's world, we're able to predict with high accuracy proteins of all difficulties. So proteins that we know a homolog of, proteins that we do not know a homolog of. For RNA, unfortunately, it's a little different. So current day, so this is results from Cas16 that along with Riju, we assessed the RNA category. And what you can see is we're kind of back in the old days of protein structure. So we have this very clear trend where once we no longer have a homolog known structure, we are very, very bad at predicting that RNA structure. So we really cannot rely on those computational tools. And one of the reasons we cannot rely on them is data is extremely limited. So this is kind of roughly the amount of protein structures that we know. And RNA is way over here with roughly 1% of the known RNA folds. So we're dealing with very little data. So our goal is to get more data and know more structures. So in our work, we think cryo-EM could be a solution to more rapidly solve RNA structures. And that's because there's many things about RNA and about cryo-EM that make them good partners. So the first thing is that you don't need to crystallize. So crystallization for RNA is very difficult. It's a skill that is very hard to learn. The other reason RNA is great for cryo-EM is it's high contrast relative to proteins. So this high contrast allows us to align very small particles. You'll see the smallest molecules that have been resolved in cryo-EM are RNA, and this is potentially one of the main reasons for that. Cryo-EM is great for RNA because it can handle heterogeneity. So many RNAs have flexible domains, and that's inherent in their function in many ways, making them hard to crystallize, but cryo-EM can kind of deal with that for us. And kind of the bonus to that is if our RNA is large enough and we're confident enough and have enough data, quality data, we're able to actually describe that heterogeneity. So we can actually start to describe multiple conformations beyond what is trapped, for example, in a crystal structure. And potentially the best part of it is it's much faster than some previous structural methods. So we need more data. We need something like a structural genomics initiative to kind of reach the levels of proteins, and cryo-EM speed certainly is attractive for that. So what is RNA? Let's go through the basic structure of RNA here. So RNA is extremely modular in structure, so it's actually a pretty good approximation to split it up into these 4 levels of structure. The first being sequence, right? So just like proteins, we have an RNA sequence, but we only have 4 nucleotides as opposed to 20 amino acids. So fewer choices, I guess. Secondary structure for RNA is simply base pairing. So that's kind of the classical AU base pairs, CG base pairs, but also some non-canonical base pairs. And you can see for RNA, we form what we call stems. So that's rows of base pairs. For example, here we have 3 main stems labeled in orange, blue, and green. And in 3 dimensions, those stems form what we call helices. So these are much larger than alpha helices, for example. We call them A-form helices. And here's just an example of what that orange stem looks like in 3 dimensions. For RNA, everything is solvated. It's a highly charged molecule. So we do not have kind of a hydrophobic core that creates globular proteins in many instances. And the shape or 3D structure is really held together by a set of tertiary motifs. So in the case of this molecule, the main tertiary motif holding it together is what we call this pseudoknot, which is these interesting base pairs here in pink and green that are kind of determining the relative orientations of these stems. And then the final level is interactions in the cell and movements, right? So for example, this RNA element is known to kind of trick the ribosome and cause a frame shift in its reading frame. And you can see here, we can also solve what the fold looks like when it's on the ribosome. So that's an important level to understand biology. All right, so what are the methods we use for RNA cryo-EM? In all honesty, it's very similar to proteins, particularly on the cryo-EM side. So the basic pipeline is we choose or design our RNA sequence. So very rarely are we actually isolating this from cells. So we're actually just choosing what sequence we want. We then synthesize it. For us, we do this all in vitro. Then for us, we always refold our RNA. Then the pipeline is very similar to protein studies. So we freeze our RNA, we image it using a Krios normally, but a glacios works as well for RNA. We process the data in very similar ways to how you would for proteins. We then model into the cryo-EM map, and then we validate our model. So I wanted just to highlight the methods that would be slightly different to kind of a protein cryo-EM single particle prep. The first is our synthesis, right? So we do this all in vitro. So very briefly, what we do is we assemble a double-stranded DNA that is a sequence of interest. We then in vitro transcribe it. So we have kind of in vitro enzymes that will read this DNA and transcribe it into RNA. And then often we'll have to purify it a little bit more. So we can do denaturing page cuts to do that. But again, because we're starting with just the double-stranded DNA of our sequence of interest, purification is a much simpler process than if you're isolating from a cell, for example. So normally we're pretty confident we have very pure sample. Folding the RNA. So in most of our studies, we are refolding it. So what that means is we heat up the RNA to denature it, and then we add salt and let it fold again, normally at 20 to 50 degrees Celsius, depending on the RNA. We can then biochemically analyze what the fold is like. So we do this through things like native gel, size exclusion chromatography. Recently, with some of our larger complexes, we've been using mass spectrometry and dynamic light scattering, but these are not so useful for small complexes. And then we go ahead and freeze our RNA. There is some slight differences in freezing RNA. So we do like using calcium-free filter paper, simply because RNA structure is highly dependent on ionic composition. So we just want to make sure we're adding the least amount of additional ions to our sample. We use very simple grids, nothing too advanced. One thing to note about grid conditions is if you don't have an additional kind of carbon film on there, you will see your RNA likes carbon compared to ice. What you'll see in a lot of our papers is we just use very high concentrations of RNA, so kind of out to 4+ mg per mil, just so the ice is concentrated enough. But you can also imagine we can use films to also get the concentration in the ice. For RNA-only samples, we do see fast evaporation very frequently. So you will see very often kind of these dry, thick grids sometimes that we think is because of the surface tension of how RNA is. We do not think RNA is hanging out on the air-water interface as much as proteins are, which actually protects it from evaporation. But that also means with RNA, so far, preferred orientation has been very rare. So generally, we see very little preferred orientation in our RNA grids, which is very convenient for us. All right. All right, so this is kind of the overview of what we traditionally do. Now, we are missing some things that in the future may be relevant. So one thing is we know that there's RNAs, such as tRNA, that are highly modified in cells, right? So they're not just the canonical A, C, G, U. They have modified bases. So this is something that when you in vitro transcribe your RNA, you're completely ignoring that effect, right? So that is a reason to do the more difficult step and isolate from cells. The other thing is by refolding our RNA, we are missing out on potential effects of co-transcriptional folding, right? So this is how the RNA folds as it's being transcribed. As with any cryo-EM study, if we're looking at the ensemble of RNA conformations, just by the act of freezing in a finite amount of time, we may be freezing out motions. And then finally, by doing this all in vitro, we don't conveniently get in situ partners to come along with our RNA, right? So if we were doing this in cells, potentially when we pull down the RNA, we may see what proteins it binds, glycans, metabolites, all this other things. But when we do it in vitro, we have to know a priori, for example, what metabolite binds this RNA and add that in. So that is another caveat with our method. All right, so let's head to the first example, which is a viral RNA. So this story started at the beginning of the coronavirus pandemic, where very early on in the pandemic, we knew the genomic sequence of SARS-CoV-2, right? So we knew that kind of first stage of RNA structure. Very quickly, we were able to predict and then experimentally validate the secondary structure of SARS-CoV-2, so the base pairing pattern of the whole genome. And one thing to note is viral RNA viruses like SARS-CoV-2 are highly structured in 2D space generally, much more structured than, for example, your RNA or human RNA would be. And the kind of Vanya and Kai-Ming started with this very interesting region of RNA, which is the first part of the genome. So this is the 5' UTR, we call it, and it contains the start codon. So the start codon is right there. And you can see it also contains very structured domains, at least in secondary space. And they wanted to ask, what is structured in 3D space? So this gets to our first step of how to choose an RNA target, right? So when you're dealing with a full 30 kilobase genome, right, we can't necessarily image the whole thing. So they decided to first focus on this region here that they thought was very structured. So SL2 to this purple SL6. They did not resolve a map that contained enough density to represent this full RNA. So then they lowered it down to just SL6 and SL5, so just this region. Again, they were not able to resolve a very nice map, so they lowered it down again, just to SL5. So this is one strategy we sometimes use to go from a very large RNA that may not itself have one 3D structure, but small domains of it may. And I just here also want to call out a warning for RNA cryo-EM in general, is that when you're solving something very large like this that has a lot of helices, those helices can all align together and you get something what we call a partial map, or something that kind of looks RNA-like, right, but is much smaller than you would expect for the RNA you put on the grid. These can be very hard to interpret and are probably an averaging of multiple of these domains, right? So I just want to caution when you see structures like this, for us, it's very critical to start to see that helical groove before we believe we're looking at something we can confidently model. All right, so starting on SL5, this is when I came into the project. What is a 3D structure we can resolve, and why are we interested in this element? Well, the first thing is it actually contains the start codon right there. So we know for translation to start happening that this element has to break apart, right? So we know at some point in its life cycle, it does change structure. It was previously proposed to contain the packaging signal, and the reason for this is if you look across coronaviruses and you look at not the stems, but the loops on top of the stems, they have this very conserved sequence. You see U, C, G, U. And this is kind of across coronaviruses, even when the rest of the SL5 element is not conserved. We consistently see this repeat unit. And even when the sequence is not conserved, this element consistently has 4 stems, right? So part of the secondary structure is conserved. So what does the structure look like? Well, it has a T shape. So we have our 4 stems, the yellow stem, blue stem, red stem, and black stem. And they are, the yellow and blue are stacked on top of each other to make a continuous helix. The red and black are stacked on top of each other also to make a continuous helix that are 90 degrees relative to each other. And in this case, based on the known secondary structure, we were able to assign what stems were which. So for example, just by measuring the length of stems, even though we cannot see the bases at this resolution, we can see there's a very short stem, 2 long stems, and we can match that with our secondary structure. The other thing we can do with RNA is we can actually decide the directionality of our strands. So just given the helical direction, we can say that at this junction, the RNA strand is moving in this direction here, so from SL5 to SL5B, and then in this direction on the other side of the junction. And an important thing to note when we're modeling this, so when we are modeling it, we know that this is low resolution. So we know we cannot be confident, for example, where we place the bases. So to try to represent this uncertainty, we propose a 10 model ensemble where these 10 models all fit in the map well, but they diverge in key regions, such as this region here, where there's essentially very little density to support where the RNA nucleotides should go. So you can see it's in red, which means the 10 models diverge a lot in this region. And you can see that across various regions that have low local resolution. So that's one way to represent our uncertainty in RNA maps, which often at these small scales, this RNA is only roughly 120 nucleotides long. We need to represent this uncertainty. Another way that we go about being more certain in our model is actually to do what we call helical extensions. And this dates back to one of the first RNA single particle structures back in 2010, where you were back then in the blobology world, right? So it was much lower resolution than this. But the general concept is if you think your stem, for example, if we think the red stem is here, and we just insert an extra extension to that stem, so we can just create that RNA that has that insertion, then in the cryo-EM map, we should also see that stem expand, right? So that's what we see from the image in B to D is we've inserted part of the helix in the red region, and we see that it is extended, confirming our placement of the red helix in that region. So this is a nice method to confirm your helical placements in cryo-EM when your resolution may be a little low. All right, so to skip to the chase, I guess, all this methodology enabled us to solve the structure of 6 different orthologs, so SL5 from 6 different coronavirus species, 4 in the beta coronavirus family and 2 in the alpha coronavirus family. And with all of these structures, the main question comes, what is structurally conserved in these RNAs? So one obvious thing we look at is the start codon position, right? So for SARS-CoV-2 and SARS-CoV-1, it's in this black stem. But then if we go to beta coronaviruses, it's in the orange stem. And then alpha coronaviruses, it's very low in the black stem. So essentially the position is not conserved. But what is conserved is those loops that had that repeat UUC GUU pattern. The distance between them is conserved. So I've just labeled them in these green bars here. And what you can see is generally the distance between the conserved loops seems to be constant. One potential problem with that hypothesis is we did observe motion in a lot of these RNAs. So for example, in the beta coronavirus SL5s, we were able to classify out 3 different structures. And you can see they are really just differing in their positioning of this down all the way to an up form. And made the choice to do this kind of heterogeneity analysis in a discrete fashion because of the size of these RNAs. The signal to noise ratio at the time was made our confidence in more advanced heterogeneity analysis methods a little too low for comfort. So we did this discretely. And we had our 3 discrete maps in this case, and we modeled into each 3 map, creating a very large ensemble of potential structures of this RNA that mainly differ in this blue helical region moving up and down, and then, for example, in this red loop where we have very low local resolution, so our confidence in the structure is very low. So what does this mean for our hypothesis that these conserved loops are equidistant throughout this whole family of RNAs? Well, we go ahead and plot this. So on the y-axis here, I'm plotting that distance. On the x-axis, you can see the various conformations of all of these RNAs. So for example, if we take the purple, and that is the MERS SL5, which we classified into 3 different conformations, we can see that the distance is equal to SARS-CoV-2 within the ensemble, right? The ensemble increases the range, but the RNA does populate a conformation where the distance is equal to that of SARS-CoV-2. One interesting experiment we got to do with this data set is we submitted some of these structures as Cas prediction targets. So kind of the best of structural predictors tried to predict the structures of these targets. And we wanted to ask, okay, if this distance is functionally important, were predictors able to consistently predict that this distance was conserved? And if we plot all of their predictions, we see some models did get the distance correct, but overall, the prediction spread was much larger than what we see in the biological ensemble. And just to hone the point that ensembles are experimentally describing our ensembles, it's important for the assessment of structure prediction. If I was to do the traditional and only deposit my highest resolution map and model for one of these structures, you can see here the best prediction is in black, and the highest resolution map in this case is in blue here. So you can see that there was an error essentially in this prediction if I was to only have deposited this model, where they essentially get this helix orientation wrong, right? But because we're looking at the full ensemble, we can actually see that this structure is much more accurate than if we were to only compare to the high resolution map, because one of our experimental structures was essentially the same as this prediction, right? At least to a very low resolution. So overall, if we had ignored ensembles, excellent predictions would have been missed, right? And this really brings me to a question when often our largest source of data is crystallography data. Which of these conformations would have been crystallized, right? And what are we actually trying to compare our predictions to? It's a very important question. So moving on now to the second example, which expands our kind of notion of what RNA structure is possible. So in the search for more RNA folds, we can take the approach of really going after something very biologically and, in the case of coronavirus, societally important. But there's also been massive searches done kind of just globally over metagenomic data. And these searches have been done in 2D space. So what happens is essentially you take the full sequence database, you do a bunch of alignments of these sequences, you look for covariation. In our case, it's base pairing covariation. And then you look for candidate RNA structural families where there seems to be covariation supporting base pairing. And this has been done quite a bit. One of the leading labs is the Breaker lab for this. And there's a whole family of labs and people involved in an effort called RFAM, which categorizes all these RNA structural families. Again, limited to 2D space, though. And they are extremely limited to 2D space because only roughly 3% of these families have 3D structures in our database, right? So there's tons of potential RNA structures here that we simply do not have 3D information on. So we went ahead and tried to look for the 3D structures of these RNAs, and we start easy. So we start with the easiest structures for cryo-EM, which actually previously would have been some of the hardest structures for a method like NMR or crystallography, which are large and complex RNAs. So this would mean they would be easier to see on our micrographs, and the complexity means they're more likely to be globular or more like what proteins we're used to solving. So this is gold RNA. So this is the largest class that's been identified in the methods I previously mentioned. And you can see kind of it's just massively complex. So I won't go through in detail, but you can see there's tons of stems. These stems are all interacting with each other, and it's just a very structurally complex RNA. It is huge. So it is roughly kind of like 8 to 10 times the size of the RNAs I was used to imaging before this. It has 2 domains that are linked by a unconserved linker region. One of those domains is quite divergent in the sequence alignment. Functionally, there's very little known about these guys. Genomically, we know they're often near tRNAs. They're found in phage and prophages, and they're found in an extremely wide array of bacterial classes. When we put these on the grid, this is some of the images we started to see, these beautiful flower-like patterns. And just for kind of context of some of the other large RNAs we're used to imaging, this is tetrahydrobinoside, which is the RNA we've been able to resolve to the highest resolution thus far. And it looks like a little globular RNA, but these are massive compared to that. And the symmetry is something we had never seen before on our grids. So if we look at the 2D classes, we can see a nice 7-fold symmetry and a nice 2-fold symmetry. And when I first saw this, I just assumed there was a contaminant because this is not something that in the RNA world we are used to seeing in natural RNAs. So symmetry and homoligomerization generally are extremely rare. So one of the highest symmetry RNAs that was previously known is this pRNA, which is the pro-head of a phage. So it forms this nice 5-mer circle here. It has just one interaction point between different chains. So it has this nice base pairing pattern between 2 chains. And it seems quite flexible in the cryo-EM data that has previously been deposited. So this guy is much larger and much more complex than that type of that pro-RNA. And once we actually collected data, once I was confident enough it wasn't a contaminant, and went ahead and solved the structure, we start to see these beautiful helices. It's clearly an RNA-only structure, and we can, again, see these nice symmetries here. So you see the 7-fold symmetry where I've just colored each chain a different color. And then if I flip it 90 degrees, you can see that 2-fold symmetry axis where you kind of have a top hat and a bottom hat that come together to form what we're calling a nanocage. And a single chain here you can see kind of comes across to interact with the chains at the bottom. So if we just look at a single chain and color it by sequence, so that 5' domain and 3' domain, so the first part of the RNA sequence and the second part, we can see that the structure of this RNA cage separates by domain. So the 5' domain and sequence space also forms the top half of the RNA complex, and the 3' domain forms this bottom ring. And now if we just look at one chain in this massive RNA and label the intermolecular interactions, we have 10 different intermolecular interaction sites. So this is a massive amount of interactions, way larger than previous RNAs that are known. Some of these interaction sites, there is evolutionary support for their interaction. So for example, some of these are base pairing interactions where we do see co-evolution of those base pairs. So it does seem like this is some evolved mechanism. If we flip it around and look in the inside, that is where that 3', 5' domain linker is. So remember that this linker between the 2 major domains of this RNA was unconserved in sequence. It also appears to be disordered. So we do not really see much density where that linker should be. So I've just drawn a connecting line here. So potentially that unconserved nature also means it does not have a dominant structure. But of note, this disordered linker is inside the cage. So if this is in the cell, that sequence would be protected from other molecules in the cell, right? So that could potentially be a very interesting functionally relevant note. I'm just going to show you some of the intermolecular interactions. For example, in B4, which is right here, it is a very classic, what we call kissing loop. So these two chains have two loops that are complementary to each other. They come together and they form nice base pairing, or a nice, in this case, a pretty long chunk of base pairing. We also have other classic interactions such as B3, which is right over here on top. And this is what we call an A minor interaction, or when a adenine from one loop comes and interacts with the minor groove of a stem. So P11, this is a nice base pairing stem. The adenine from the loop of loop 9, we're calling it, comes in and interacts with that, those base pairs. And then we have some kind of more non-canonical or non-traditional interactions that are mostly driven by hydrogen bonds between different bases. So this is a really interesting one on top here, where the loop of stem 7 is interacting with the stem of stem 7 from a different chain. So this is what allows the top to kind of close together and form kind of the top of the cage. Very interestingly, we also have another family that we looked at that also appeared to be forming these cages. And if you look at the 2D classes, the symmetry seems to be different. So we have a twofold symmetry axis, but now we have a fourfold symmetry axis that's the top view as opposed to sevenfold. This RNA family, there was no reason to believe that they would be similar in structure, but they do have a similar genomic context. So it is roughly half the size, one monomer. It is also often near tRNAs. It's found in phages and prophages, and found in also a pretty diverse set of bacterial classes. An interesting note about this RNA is it can be highly expressed in some species. So one study that looked at a lactose species showed that after 24 hours of growth, the expression of this RNA in black here was equal or near equal to the expression of 16S RNA or ribosomal RNA, right? So this RNA is getting produced in massive amounts, essentially. And again, just like gold, the two domains are linked by an unconserved linker. And this linker, when we solve the structure, is also inside the cage. Based on the secondary structure, there was no reason to believe these would be a conserved three-dimensional structure. But it turns out they are, and there's some sort of convergent evolution here for both of these sequences to make nanocages. I won't have time to discuss some other structural classes that we were able to solve with cryo-EM, but based on going through classes that looked interesting in 2D space, we were able to show quite a few of them resolve in three-dimensional space as well into these very atomically ordered RNA structures. And just I want to call out one of these examples before we move to questions. That is the Ole RNA. This is an RNA I actually included as a negative control. Negative control because it was a known member of an RNA protein complex. So traditionally, when we think of, for example, the spliceosome, when we think of that RNA in the RNA protein complex, the RNA is very flexible and is scaffolded by proteins. So I assumed if I went ahead and kind of solved the Ole RNA without proteins, it would be a very flexible RNA. But it turns out that the RNA is actually highly ordered by itself without any of its protein partners, indicating that maybe our common wisdom may be a little premature or incorrect in some instances, that maybe in this case, the RNA is actually scaffolding the complex. So I think given these complex folds that we're really only able to reveal now because of cryo-EM and the lack of crystallography necessary to do cryo-EM, we may be entering a new world into where RNA is actually a scaffold, either for itself, such as the golden rule, or for proteins in the oRNA case. So the role of RNA kind of in structural biology, maybe we have to relook at our assumptions that it's super flexible and think of it as a potential scaffold for structures as well. So what have I gone through today? Well, just to introduce the importance of RNA biology, we cannot rely on the predictions of computational methods for structural information, and we really require more RNA structures, and we believe our cryo-EM may be the place to look for that. Cryo-EM is fast, and generally once you find a grid condition that works for one RNA in a family, it will tend to work for other similar molecules. So this enabled the study of many more orthologs of coronavirus SO5 RNA than reasonably could have been done before the era of cryo-EM. It can also describe multiple conformations, which was important to confirm conserved structural features within an ensemble. And it also enables a more fair assessment of prediction accuracy. And cryo-EM in general has enabled a higher throughput screening of RNA tertiary structure. It's still not high throughput, but certainly higher throughput than previous methods. And this has enabled the discovery of these very large homoligomeric RNAs, where RNA-RNA interactions scaffold the structure. Want to acknowledge, obviously, this talk is all about my cryo-EM, so S2C2 has been the source of the majority of my cryo-EM time. I also started with absolutely no training in any of this, so a lot of the people in S2C2 are integral to my training and my ability now to solve these types of structures. So thank you so much, S2C2, for being so accommodating to me. The True Lab and the DOS Lab is where I do my research. They have been obviously very helpful in forming questions on RNA biology and what is possible with cryo-EM. And then on the SL5 project, we have specifically Lily, which did a lot of the ortholog work, Vanya Kaming and Shan that did the initial studies into what region we should target for cryo-EM, and then Zuting, Ray, and Grace that helped with some of the cryo-EM screening for all these structures. And in the ornate RNA project, I'd like to thank Vivian, which did a lot of the synthesis of these RNAs. And then I did not have a chance to talk about their work, but the Gao Lab and Kunin Lab were integral at looking at evolutionary support for those intermolecular interactions. All right, so I think we have time for questions, hopefully. I'm happy to take any and all.
Thank you very much, Rachel. That was absolutely incredible.
We have a bunch of questions. Some I think will be easier to answer than others. The first one is, why do you denature your RNA if it's already folded before refolding it?
Yeah, so we essentially want to make sure that it's in a consistent state. So if we denature them all at the same time and refold them all at the same time, now we know the majority of our molecules are going to be in a similar folded state. The other reason is when we are doing in vitro transcription, we are using a viral transcriptase, that the transcriptional rate may be different from our native transcriptional rate. So we may actually not have the native co-transcriptionally folded RNA anyway. So.
Thank you. The next question is, does the electron density have enough features to distinguish an AUCG sequence?
Yes, at certain resolutions. So the difference between an A and a G, you can start to see at roughly 2.5 angstroms. The difference between a U and a C is much harder to see. that is probably going to be closer to 1.4, but we're simply not at that resolution of RNA-only cryo-EM to do that. But the difference between a pyrimidine and purine, you can see very early on. So something like 3.8 angstroms often, you can start to see that. So just based on that and knowing the exact sequence in your RNA, you can already map out the sequence.
The next question is the cage of gold known to occur in vivo?
Unknown as now. So all of this work obviously was done in vitro. We did confirm that at concentrations that it would be in the cell, it would form. So we did some mass photometry and dynamic light scattering at concentrations comparable to cell expression, and it does form. But obviously the cell is much more complex than just a low concentration. So unknown for now.
And a related question to that is, do you have any thoughts on the natural biological function of gold, or ROOL, the rule?
Yeah, there's tons. So these nanocages are essentially what often is called microcompartments in protein world. So those can have a huge variety of functions. So they can encapsulate things to remove them from kind of the cytoplasm. They can store stress proteins, and then the cage kind of opens up when stress occurs and releases those stress proteins so they can respond very fast. So there's a lot of different potential functions. I think in many ways we think the cage structure probably is important for whatever function it's having. So for some reason, for the function, it's useful to have things separated from the cytoplasm. But what exactly that function is, it's a very wide array of hypotheses right now.
Cool. The next question is dealing with contrast. And from the questioner's perspective, many of the RNA maps, the strongest signals are coming from the base pair interactions and not necessarily the phosphate backbone. And I'm wondering if you had an explanation for this, or thoughts on why you get more contrast from the base pair and not the highly charged backbone.
Yes. So there's a very interesting effect with base pairs. At roughly 3.5 angstrom resolution, you start to see, so if you're below 3.5, your bases are just all blurred out. Once you get just slightly above that, you have beautiful base separation. And you see essentially a very steep trend with kind of the quality of your RNA model when you go between those kind of resolutions. So that's one phenomena. The thing about backbone is the reason as similar resolution sometimes is not as well resolved, so this is talking about a 3-ish angstrom resolution, is because it does have a phosphorus, so that is good contrast, but it also has negatively charged ions. Oxygens, negatively charged oxygens. So those can contribute negatively to the overall signal. So the phosphorus and those oxygens are kind of combating each other. And that is why in certain resolution ranges, you probably see not as great backbone density as you would expect given the phosphorus.
Thank you. Let's see here. For the next question, let's see, we have a bunch to pick from here. Could these methods be applied to very highly dynamic nucleic acids?
In theory. So when you're talking about highly dynamic nucleic acids, you have a battle here. And the battle is partially with size. So the smaller it is, the harder it is to determine the orientation of your molecule. So whether it was frozen in a top view or a side view, for example. And then when you're trying to differentiate conformations, because you're unconfident in whether it was a top or a side view, now you're getting orientation and conformation all mixed up. So I'd say for dynamic RNAs that are very large, I think we're already at the point that we can address those. But for small ones, I think we're going to have to get either improvements in engineering to get better signal to noise in our data, or improvements in the current computational methods to address that uncertainty.
Mhm. We have a couple different questions about the RNA prep process and the refolding. One is, do you do all your RNA refolding in the exact same buffer conditions, or is it very molecule specific?
Yeah, that's a good question. So our default for the lab is to do 50 millimolar sodium Hebe's, pH 8, and 10 millimolar magnesium. That's default. Um, Obviously, there are some RNAs where we know they need potassium. So something like a G quadruplex, we'd add in potassium. Um, Also, that high magnesium sometimes causes aggregation. So sometimes we actually, if we know our RNA still folds at a lower magnesium concentration, we'll on purpose lower that magnesium concentration so that we reduce aggregation. And all of that kind of folding magic, I guess, can be RNA specific, and that is why these biochemical analysis tools, like a native gel, for example, are important for kind of testing out those folding mechanisms. But I would say our default is just to do our normal buffer, look what it looks like, and go back to the drawing board if it appears aggregated or unfolded.
I think you've also answered a question about your basic purification scheme after you have done your in vitro transcription. So I think I'll mark that one also as answered. Let's see here. How are we doing on time?
Yeah, so we do have a few questions still. Rachel, if you can hang on for like 5 extra minutes, if that's all right with you,
Oh,
I can stick around and we'll get to those questions. For people who have to pop off, we'll keep recording. So if we didn't get to your question and you have to leave, come back to the recording to hear. We'll try to get as many of these done as possible.
Uh, so another question is about particle counts and the number of particles you typically find in your reconstructions of your viral RNAs.
yeah. So for viral RNAs, it's a lot because these guys are relatively flexible. So, what I said in the start about three-dimensional structure being kind of dictated by these tertiary interactions, most of these viral RNAs are just four helices and don't really have anything keeping them super tight together or in a defined form. So that's the reason that they're pretty low resolution, and that's also the reason why you need a lot more data than you would for something that's highly ordered or globular. So for those, it's on the order of 1 million particles for each of those reconstructions. Once you try to dissect motion or flexibility or heterogeneity, you need to multiply that by a factor, right? So if you have three conformations, generally that would be in the traditional method of heterogeneity analysis, 3 million now, roughly.
Related to that question is, what do you think limits the resolution that you can achieve for your RNA cryo-EM structures? I mean, it clearly
Yeah.
can be a combination of factors, but do you feel anyone dominates more in your experience, or
Um, Yeah, it's a little hard to say, right? Unless we systematically test this, which we haven't. Um, I'd say just from experience, the main one kind of just biophysically that seems to be dictating it is simply motion. So, for example, those four helices, right? You can imagine the ends of them can drift, and that causes very low resolution. If we were to focus on the junction, for example, it would resolve better if it was larger. The other consideration for RNA, which I think is maybe a little unique, is what I call repeat units, or more traditionally, I guess something like pseudosymmetry, you would call it. And there's starting to be computational methods to address this, but there can be more. And that's simply the fact that, RNA is just a bunch of helices, and one helix looks like another helix, right? So when you're doing your alignments, and if you have a relatively simplistic RNA, such as we have H-type RNAs that are literally just shaped like an H, you can imagine there's some pretty obvious pseudosymmetries there that can cause problems. So I'd say flexibility and pseudosymmetries are the main problem for these very small RNAs. But it has not been systematically tested, so hopefully someone will do that soon, and then we'll know for sure. But
Okay. Another question is you've explained the advantages for cryo-EM for RNA structure very well. Do you see value in pursuing very high resolution crystal structures for very small RNA fragments, and using the cryo-EM, what we learn from cryo-EM to help us pick those tiny fragments?
yes, 100%. So I'm a big proponent of kind of using multiple methods and learning what you can from each method. So RNA is great for global fold, right? Particularly of these flexible RNAs. Something like NMR or crystallography, for example, in the viral example I showed, right? If we think the loop structure is really important, then we can isolate that loop, just create crystals or use NMR to solve kind of the ensemble of loop structures it could have. And now that combined with kind of the global fold we're able to see from cryo-EM, we can now really start targeting, for example, drug targeting, right? Which with low resolution cryo-EM is not necessarily that informative. So I think that is the way a lot of these flexible or low resolution RNA cryo-EM maps should go if we want to get more atomic level resolution. But large RNAs you can already solve to atomic level resolution with cryo-EM, so those ones maybe just stick with cryo-EM for now.
Related to that and large RNA structures, this next question is asking if it would be possible to obtain a structure of an RNA G quadruplex in the future, for example.
Yeah, there are maps that contain G4s or G quadruplexes. And the exact publications are escaping my mind right now, but they do, it has been done. They're quite low resolution, right? So, for example, you don't see the separation of the stacking of the Gs. But you can use crystal structures, going back to the other answer, of those G quadruplexes and place them in the density and then get a better idea of the global structure of your full thing with the G quadruplex. So certainly possible and has been done. Yeah.
Cool. Our next question is related to the extensive information coming out of the Breaker lab that you had mentioned, where they've biochemically characterized many of the large non-coding RNAs, but they've yet to determine a biological role. Do your structures or the biochemistry you've done point to any functions in cells of viruses for these?
Yeah, I think in many ways this study was seen as a hypothesis generation study, right? So I think, for example, the nanocages, right? Simply having a compartment leads you to certain hypotheses that down the road can be tested by hopefully a lot of labs in the field to try to figure out what these guys are doing. I'd say because there was no homology structurally to anything else in the database, there was no obvious hit, right? It wasn't like it was exactly shaped like a known ribozyme, for example, and then we know exactly what it does. But it generates a lot of hypotheses for that. Similar of the OLE. So while we did not solve it with proteins, protein binding domains that were known are solvent exposed, and it's very obvious where the proteins are supposed to go. So because of that, now we can start generating hypotheses on how the proteins are orientated to each other and how they may be interacting, for example. But again, did not lead to any clear functional information without doing further biochemistry that we have not done ourselves yet.
Excellent. The next question is also related to the cages, and what buffer conditions lead to their formation and assembly, and if that provides any insight into their mechanism for assembly and disassembly.
Yeah, these guys seem to just assemble, essentially, to put it simply. So with just a few of the sequences, we haven't done like a cross-family analysis of this, but they can fold in very low magnesium conditions, so it doesn't seem like magnesium is necessarily super important. They can fold without potassium. So it seems like just because of the raw number of interactions, they can fold under pretty mild conditions. They are also quite stable. So the complex doesn't fall apart till roughly 60 C, 65 C, depending on the RNA you're looking at. So it also seems quite thermostable. Yeah. Hopefully I answered it.
Okay. The next question is, at what resolution would you consider a structure as, quote, solved? For instance, the viral RNAs at 4.5 to 6 angstrom resolution, it's not clear which kind of reactions hold the tertiary structure together with regard to unusual base pairing or hydrogen bonding to phosphate groups, etc.
Yeah, I guess this is kind of a philosophy question. So in kind of how I view things, I'd say the structure is never going to be solved because in every experiment, right, we are perturbing it in some way, right? So in cryo-EM, we are freezing on the millisecond timescale, right? So if we're trying to describe the ensemble or the conformational landscape of the RNA, we know we are already distorting that. So I'd say the goal in these kind of structure determinations, as opposed to solving the structure, is to outline what parts of the structure we can be confident in, right? So, for example, for the viral RNAs, we know for two of them that there is a tertiary interaction there, but it was not resolved in sufficient resolution for us to tell you what it was, right? So we did not make any major conclusions off of that. But the loop positioning is something we can't see at that type of resolution, right? So I think it's less of a claiming it's solved and more of a knowing what information you can confidently deduce from structures at given resolutions. There's some philosophy for today, I guess.
Mm-hmm. So we have 3 questions related to Gold's RNA structures. One is, do you see any modifications of nucleotides? And
Okay.
then and do all of them form 14 mers, or are there other multimers like decamers?
Yeah. Okay. So modifications, since we didn't isolate from cells, I can't say if there's any natural modifications. In our in vitro system, there wasn't, of course, because we don't put any modified nucleotides in the test tube. So that one, unfortunately, I can't tell you an answer for. For the oligomeric states, in the structure I showed, it really only forms that one stoichiometry. There are some golds which we have looked at that don't have a 5' domain, so they don't have the top and the bottom of the cage. They just are that kind of center ring. And those guys can be a little bit more flexible in their stoichiometry. So, for example, they can be a 14 mer or a 12 mer, but they don't show kind of a massive spread in that. And those we think potentially it's just because we looked at them without the 5' domain, and the 5' domain may be important in determining the exact stoichiometry. Hopefully that answered the curiosity.
Related along to that curiosity, based on your work with golden rule, are there hints of other similar oligomeric RNA complexes that might exist in your eukaryotic cells that you're on your radar?
Yeah, I would love if there was more. So I think from the RNA families and two-dimensional structure space that have been discovered so far, these are certainly the largest, right? So I think if we're looking at those families, it's going to be smaller. That doesn't mean that there's not families that have not been discovered yet, right? So there totally could be. And I think by revealing the intricacies of these intermolecular interactions, we're starting to get a set of signals to search for, right? So, for example, these structures have self-interacting loops. So what that means is like loop 7 from chain A interacts with loop 7 from chain B, right? And normally when we're not thinking about RNA-RNA interactions, we would completely ignore that because two nucleotides right next to each other cannot interact, right? So that type of signal, if we do see covariation in kind of the same loop, that could be a signal for these type of interactions. Generally, we've ignored that till now, right? Because we didn't know to look. So potentially in the future, people will start looking and may find more. But right now, we do not know of any more. Yeah.
Cool. And then our last question for you here is that their RNA is forming aggregation towards the periphery of grid holes. And if you could suggest any tricks to avoid that, and if there's any reasons for aggregation, things that might be causing the aggregation.
So aggregation at the kind of, I guess, carbon water interface. I don't know what the proper name for it is. We see quite a bit. So it's not rare, let's put it that way. We especially see it with RNAs that are less structured. Like the viral RNAs, for example. So potential ways to mitigate the aggregation. One is potentially you're folding at too high of an RNA concentration. Depending on how you're folding, obviously. So for us, if we fold at too high of an RNA concentration, there's just too many RNA molecules in the same tube. They interact with each other and start to aggregate, right? So how you can avoid that is fold it at a lower RNA concentration, concentrate it down, and then freeze it. The other possibility is, I think I mentioned this before, is magnesium concentration does also affect aggregation. So lowering your salt concentration and seeing if that helps. And then the third thing is just to change your grid conditions. So if you have a carbon film or if you have a graphene film over top of all your holes, you need to freeze at a much lower concentration then, sometimes as low as 10x what you would if it's just a water hole. And what that means is you're going to reduce aggregation as well, right? So those are things to try. At the end of the day, not all of them may work, but those are the things generally we try. Yeah.
Excellent. Thank you for your presentation, Rachel.
Yeah, thank you so much, and thanks for sticking around to get to all those questions. There's still a number of people here, which means this was extremely useful information for our community. So thanks everyone for coming. Join us again next month. We will not be talking about RNA, but hopefully something just as beneficial. So I'll go ahead and stop recording.